Hello, this is Michael Anderson, Teresa Ha, and Angel Rojas, and this is our presentation growth analysis of Neochloris, Oleoabundans, and Wastewater. So algae has been claimed to be a potential multi-beneficial problem solver for some of our more modern issues. The algal strain can be synthesized into biofuels, made into bioplastic, used for medicinal purposes, utilized in the wastewater management process, made into animal feed, converted into dietary substances, and used for cosmetics and dyes. Furthermore, algae can be used to kind of mitigate the global warming crisis by retaining the CO2 until it's time for combustion. So instead, what we do right now is we just take things that are already retaining CO2 and burning them up, releasing the CO2. We'll actually retain the CO2 ourselves and then burn the CO2. Some of the opponents of algae for biofuel have pointed out that committed land masses could decrease, such as the committed land masses used for food. They feel like that might be a competition, but the proponents are pointing out that algae can grow on the surface of water, and it really doesn't need that much, that large of a quantity of committed land masses. So our algal strain is named Neochloris oleoabundans, or NO, which is what we'll be calling it because it's a lot easier to say. Our algae is in the class Chlorophyceae. Strain is generally considered a great candidate for feedstock, biofuel production, and cosmetics due to the high lipid content in comparison to other strains. This lipid content is due to it naturally occurring in a desert environment where water and nutrients are scarce. This particular algae was obtained by the UTEX culture collection of algae. So the bioreactor conditions. Growth medium, it's filtered from the Fresno Basin water. It was collected early February 2021. Had 8 days of continuous light right there with these LED strips you can see. Any portion of NO culture was removed, then it was replaced with equal volumes of fresh media. Bioreactor was cleaned out on the 8th day.
The experiments that were done during this project were run in 3 trials, and the best data from those 3 trials were selected for this presentation. The following data in particular will be shared in this presentation, and they include the culture growth, nutrient removal, lipid accumulation, FAME yields, and protein analysis. Now, culture growth. We observed an upward trend when we graphed cell counts versus absorbance, and we did it as a function of time and days. We were able to observe this trend using a hemocytometer, and the computer picture to the right is an example of a hemocytometer, and it shows how we're able to count the cells and what areas specifically to count them in for statistic quantities. Here we've made the assumption that day 0 has 0 cells. However, this is not actually the case. Nutrient removal. We look to the graph on the right. The nutrient is in blue and the phosphate is in red. The NO algae structure needs phosphates and nitrates to grow, and thus we expect these concentrations to decrease as the algae cells remove them from the basin water medium. Day 7 in particular shows an outlier for a phosphate concentration that's a little higher than usual, and we believe that this is possibly due to contamination. Now, lipid accumulation, and we look to the right on the TLC plate, the TLC plate was prepared utilizing a tube mobile phase that moved the different contents to different links in the superior direction of the TLC plate. And this was measured and observed under UV light after we have separated the lipids. The plate yielded spots and rough placements, but identified from top to bottom as FAME, TAG, FFA, DAG, and MAG. Each of these are used in synthesizing alternatives to to diesel fuel. Now, FAME, otherwise known as fatty acid methyl esters, the FAME extract was dried and weighed for yield after a number of isolation steps. What we've got from the results is about 0.0203 grams of FAME extract with a reported biomass of about 13.92%. Other studies have shown that when efficiency is maximized, the NO strain can reach up to 37% yield. This is one of the more beneficial traits of this algal strain. An example of the algal strain is in that picture on the bottom right in that falcon tube.
To determine the protein concentration, the DC protein assay was used. When proteins react with a Folin reagent and alkaline copper tartrate, they create a blue-colored solution. This can be read by a spectrophotometer since it absorbs light. If we create a standard curve with gamma globulin, we can determine the concentration of protein in an unknown sample. From our experiment, we determined that our sample of NO showed a 23.8% concentration of protein. According to other research, the median concentration is about 31%.
All right, so special thanks to Dr. Kimulani Mitra and Alina Song for providing help and assistance during our experiment. And that's it. Thank you.